All right, so we're going to talk about phylogenetic trees in more detail today. If you have your clickers, please get them out. In about 10 or 15 minutes, we're going to do a test to try and see whose clickers are working, and we're going to try and get things up and running in time for tonight's class. So we'll get to that in a few minutes. All right, so just as a reminder, or for those people who weren't here, in the previous lecture, when we weren't talking about administrative details, we talked about phylogenetic trees and some of the parts of a tree, and then the big picture of where we're going in this course in terms of the tree of life. And what we're going to do today for this lecture is talk about phylogenetic trees in more detail, the structure of a tree, how to move around a tree in different ways, and then some of the groupings of organisms in trees, sort of an important terminology as well as ways of thinking about the groups of items in a phylogenetic tree. And then in the next lecture later today, we're going to talk about, rather than organisms, actually tracing the evolution of features of those organisms given a particular tree. So we're going to start relatively simple here, but in fact, a lot of the stuff we're doing today is relatively simple, but it's really, really important that you're able to understand the rotation of trees and the different components of a tree and the different terminology for groups on trees for the rest of the course. So we're going to try and go through this maybe in too much detail for some of you, but hopefully by the end of today, everybody will have a good feeling for just what a tree is showing you. So the first thing we're going to talk about is rotating phylogenetic trees and one particular aspect of rotating. So if you have a piece of paper, please draw this tree. We're going to use it for a couple of different things for today's class. It's also in the lecture notes if you already got the slides. In a tree like this, it's called a vertical tree. I've said this already before, but I'm going to go through it again. A vertical tree, the time axis is the y-axis. So the elapsing of time goes from the bottom to the top, from the past to the present. And this vertical tree, if you have it on a piece of paper or if you have an iPad or your lecture notes, you can just pivot it 90 degrees, rotate it 90 degrees, and you get a horizontal tree. All we've done is rotate the piece of paper. We haven't changed any of the meaning of anything in the tree. The time axis now changes to the x-axis, and it's now called a horizontal tree. But it has the exact same meaning as a vertical tree. And we're going to go back and forth in lecture. We might show you a vertical tree, a horizontal tree. We might show them back and forth for the same group of organisms. You have to be able to just rotate them in your head and understand what they mean. And what we're going to go through today is sort of other ways to think about the components of a tree. So we can do the same with a real tree. I'm going to be using mostly sort of these hypothetical trees today, but we can do the same with a real tree. Here's the chimpanzee, human, gorilla, orangutan tree. And we can take it and rotate it 90 degrees and go from a horizontal tree to a vertical tree. And again, if you just look at the structure of the tree, the branching order, the branch lengths, the patterns in the tree haven't changed. We've just changed the positioning in the tree. So that's a sort of straightforward looking at rotation in the tree. In a minute, we're going to come back to rotating individual branches in a tree, which is a little more complex, and you have to be able to wrap your brains around that. So what I want to do before doing that is just go through the different components of a phylogenetic tree, again using this same tree, hypothetical tree here to think about the different parts of a tree. So who knows what those are labeled in yellow on the tree? Yeah, so they're the taxa in the tree. And taxa is plural for taxon, and taxon is any individual organism or group of organisms that we designate a name for. So it could be a species, and we give it a name. It could be a phylum, and we give it a name. And in this case, this tree was meant to represent the evolution of species, but it could just as easily have been the taxa at the tips of this tree could have been orders. of birds or families of some other group of organisms. And in every case, we're showing the phylogeny, the evolutionary relationships of a set of taxa that we're interested in. And taxa is just plural for taxon. So what are these? So those are the nodes in the tree. And I've mentioned this before, but we're going to go through it again. So there are two ways of thinking about nodes, really. If we start in the past and we wander our way along a tree, so you have some ancestral lineage represented by a branch, and you get to a point where that lineage, say it's a population of humans or a population of bunny rabbits or whatever, splits into two distinct lineages, maybe because a mountain chain came up between them, or they moved one, rafted to some other place, a population of them. Whatever the mechanism, which was discussed in BIS2B for mechanisms of speciation, we're not going to focus on that here. What we care about is that they've now split into two distinct populations. And those are now represented by two, they each get their own branch. So coming out of a node, you have two branches emerging. And those represent the two lineages that come from a single ancestral lineage. So it's a split in the evolution of that particular lineage. If you think about this going backwards in time, from the present to the past, a node also represents the most recent common ancestor of a particular group of organisms. So if we look at A and B, their most recent common ancestor is represented at this node labeled F. And I'm being very specific here, saying most recent common ancestor as opposed to just recent common ancestor, because anywhere along the branch leading up to F are common ancestors of A and B. Those are all common ancestors of A and B. The node at which the split occurs to lead to A and B is the point at which the most recent common ancestor existed for those two taxa. So again, node means the same thing, but it's just in your head. If you're going from the present to the past, you might think about it a little differently than if you're going from the past into the present. So we actually have multiple terms for different kinds of nodes. And I'm just labeling some of them here. So does anybody know what these nodes are called? Yeah, so these are internal nodes. They're on the inside part of the tree, so they're called internal nodes. I'm not labeling them, but the taxa here are sometimes also called terminal nodes. We're not going to use that term probably anywhere in the class, so I haven't labeled it or talked about it. And then there's one special other node in the tree. Anybody know what that's called? Yeah, so it's the root node, not the root, and we'll come back to that in a second. So this is the root node, and it represents the most recent common ancestor of all of the taxa that are shown in the tree. Or again, thinking from the past into the present, the point of the earliest evolutionary split for the ancestry of the groups being studied. So please also, anybody has any questions, just holler or throw things at me, whatever, along the way. So, anybody know what do you have a question? Oh, sorry. Fidgeting. What are these called? The branches. And just like, well, I'll get back to them in a second. So branches represent the evolution of taxa over time. It's very important that you realize that this is really sort of a clock-like behavior of the clicking of time occurring. We are not trying to represent with this particular tree any type of change in the features of an organism. We will come to that in the next lecture. So this is just representing really time elapsing as a lineage, is reproducing, existing over time. If an organism or a lineage goes extinct, we stop the branch at the point where it goes extinct. It's really just representing time, not features. Just like with nodes, we have different kinds of branches. We have internal branches on the inside. We have terminal branches going up to the tips. and we have one special branch called the root, or the root of the tree, or the root branch. And the root branch, or the root, is very important for many purposes, as we will see certainly in the next week or so. It's the branch leading up to the root node, and it's the branch leading up to the most recent common ancestor of all the taxa in the tree. And along that root branch, anywhere along that branch, exist common ancestors of everything that you are looking at on the tree. Again, when it gets up to the point of the root node, those are the most recent common ancestors of everything in the tree. So I think this is included in the slides. I've made a little figure showing all of these. You should understand these terms, know how to label a tree like this, and most importantly, we're going to refer to these terms, in the middle of lecture. We're going to talk about terminal nodes and terminal branches and the root and root nodes, etc. You have to understand what those are. It's not particularly complicated, but you just have to get comfortable with that terminology. Are there any questions about this sort of structure of a tree before we move on to something else? Okay. Yeah, sorry.
Are the MRCAs, are they still called taxa?
Oh, yeah, great question. So are the MRCAs still called taxa? So if someone gives something, tries to assign something a name of an organism that occurred in the past, then it becomes a taxa or taxon. If it's just sort of this nebulous existing entity somewhere in the past, if people are being really technical, they might not call that a taxa or taxon until it's been sort of formally given some sort of recognition with a name. So I wouldn't worry about the exact details there. In most cases, when a tree has particular points labeled on it in the past, either a node in the past, representing the most recent common ancestor, or individuals along the branches, like if you have a fossil of something from the past, we can call those taxa. So they're entities that we are specifically interested in the evolution of, and we'll just refer to them as taxa, or the individuals as a taxon. Any other questions about the structure of a tree? Okay. So now, once we have that sort of structure of a tree, the important part in that tree, I'm just going to go back to this slide again. The important part in this tree is the order of branching in the tree and the groupings of taxa by their most recent common ancestor. So A and B have a most recent common ancestor here. A and B together with C have a most recent common ancestor here. We could have had a tree where C was somewhere with D, say on the branch over here, and then C and D would share a most recent common ancestor. Even if we drew the tree with the letters in the same order, so C could have sat in the same point on this drawing of a tree, and I could have switched the branches such that C and D were grouped together. So the order of these taxa drawn on this tree has not changed, but the branching pattern has changed. That's what you need to focus on, is the branching patterns. That's what tells us the evolutionary history of the organisms in the tree. The order of the labels from left to right on the tree has no meaning. And to see this in more detail, I'm going to show you what happens when you rotate branches on a tree. We're not changing any of the branch order. We're not changing the position of any of the nodes. We're not changing the meaning of any of the tree. But we are spinning branches around a node. So you should imagine that an evolutionary tree is sort of like a mobile, hanging with each branch and then a little ball holding together the branches that hang off of it. Or a chemistry stick, ball model, if you are obsessed with those right now and taking chemistry. And you can spin the ball that represents the node any way you want. And you have not changed the meaning of the tree. You can't unplug a branch from a node and plug it into another place. That changes the meaning of the tree. But if all you're doing is spinning the node around, you haven't changed any meaning in the tree. And so I'm going to show you this. So again, take this same tree, and we're going to rotate one of the branches. So let's take this branch over here with G and H together and rotate it around. We're spinning that node around in the tree, and now H is on the left and G is on the right. The meaning is identical. We have not changed the meaning at all of this phylogenetic tree. G and H share a most recent common ancestor with each other to the exclusion of everything else. That is the only important part of G and H for this particular rotation that I did. So you have to be able to do these rotations in your head, because we will show you trees and expect you to understand what the meaning is of the tree, regardless of whether or not G and H are on the left or right, or any of the other taxa in the tree. So again, a little abstract maybe with that tree, but if you look at these two trees here, they mean exactly the same thing. We've now taken a vertical tree and rotated it, so this is a horizontal tree, and we have spun around one of the nodes between the two trees. So this node here that represents the common ancestor of humans and chimps, we've spun it around so that in one of the trees chimps are drawn on the top, and in the other tree humans are drawn on the top, but they mean the same thing. Humans and chimps share a most recent common ancestor to the exclusion of everything else in the tree. The branching order, also known as the topology in the tree, has not changed at all between these two trees. So you have to be able to do these rotations really for all the branches in the tree and be able to understand what the meaning is. Any questions about that? Okay, we're going to try a clicker question. So if you can get out your clickers, I'm going to start the system. I'm going to see how many glitches we have. And I've labeled this, I think, as it should be showing up as BIS2C if you're looking at the identification. Yeah.
Is this something we're supposed to register online? So we got this like 10, 20 minutes.
Yeah, so we're not this is practice. So we're trying to see if it will communicate with the system. For actually getting points and registering bonus points, you will have to register it online so that we can identify your student ID from the code on the iClicker. So try and do that before tonight's class. But this is not to count. This is just for everybody to see if it is working for them, and then we can try and kick out the bugs for the people where it's not working. Okay, so here's the question. Consider we'll get to questions about the clickers in a second. Just let me read the question. Consider the following trees, tree 1, tree 2, and tree 3. Which of the following statements is true? Tree 1 is the same as tree 2. That is, it means the same thing. Tree 1 it means the same thing as tree 3. All three trees mean the same thing, or all three trees are different. And when we do clicker questions, you're welcome to talk to your neighbors to try and work out the answers. We'd rather you didn't, post things to Facebook or Twitter while you're trying to solve the answer, but a little local environment and social interactions is fine. So you have a question about the clicker system?
Yes, which frequency?
It should be on frequency AA if it's not showing up to you. With the iClicker system,
It shows AA when you start it up, right?
yeah, it should show AA when you start up your clicker. Yeah. Do you have an iClicker 2 or a 1?
2.
No, the one who wasn't working.
Oh no, I just don't know which frequency it was.
And does it work now if you select the AA?
I can't tell. What would it say?
It should, when you send in an answer, it should say received or...
It's not telling you to see.
Turn it on and off and see. It should say AA when it turns on. So, can we just have a raise of hands of how many people think your clicker is not working correctly? And how many of you have, of those people, do people have the iClicker 2 or the iClicker 1? You have the 2 and it doesn't seem to be working? All right, well, for the people who it's not working, if you can see me after class, we will try and figure out what's going on. But at least many people have answered because we've gotten 253, answers so far. And most people have answered A, which is good, because that's the correct answer. All right, so the way I would approach answering a question like this, the way I do it in my head, there are two sort of ways that I approach a question for comparing the topology of two trees. One of them is I look at the groupings in the tree, and I compare whether or not I can find the same exact groupings in the multiple trees. So U and V, let's just take tree number 1, and there's a grouping here of U and V that share a common ancestor. We can find U and V together in the same way in tree 2. Now, V is on the left and U is on the right, but that means the same thing. They're grouped together. That's all that matters. On the other tree, on tree 3, U and V are not grouped together in the same way. V is actually shares a most recent common ancestor with W to the exclusion of U. We can't draw a little circle around U and V in the same way we can for tree number 1 and tree number 2. So tree number 3 is topologically different than tree number 1 and tree number 2. It means something different in terms of the evolutionary history of the organisms. So we're going to throw that one out and then keep comparing the trees. So tree number 1, the group of U and V together shares a most recent common ancestor with W. It does that in tree number 2. Z and Y are together in tree number 1. Z and Y are together in tree number 2. X is together with the group of Z and Y in both trees. So the two trees are the same. That is, the order of branching is the same between the two trees. So I usually do it by looking at these groupings. Another way that's basically the same thing, but another way to do it is to just look at every node in the tree and compare the taxa that are descendants from that node and see if you get the same collection of taxa in the two trees. So at this node, the descendants of that node are U, V, W, X, Y, and Z. That's the same in all three trees. So for the bottom node, they're all the same. Here's another node, U, V, and W, U, V, and W, U, V, and W. So even tree 3 has the same descendants from that node. But if we go up to this node, now we have U and V. Can we find a node like that in the other tree? There's U and V here, and we can't find a node like that in tree number 3. So they're different. A third way to do this is to actually rotate the branches in your head between the trees and try and see if you can rotate them to get them to be equivalent. As you get bigger and bigger trees, that is a nightmare. So I would suggest trying to do it with the groupings here. Maybe as a last final check, try and do some rotation in your head or on a piece of paper. But it's the groupings of taxa that are the key to figuring out if two trees are identical in meaning. So what I want to do now is actually take you through a little bit more detail on terminology and ways that groupings in trees are important and the ways we think about this. So I'm not going to give a lot of examples right now, but this may seem like a bit of an esoteric exercise to people, but it isn't. There are many, many reasons why you would like to know, for example, if two samples or taxa that you are studying share a most recent common ancestor with each other to the exclusion of everything else that you're studying. So are these two taxa grouping together to the exclusion of everything else? And I'm just going to give you one example of this, which relates to HIV, the HIV virus. So it turns out that when people have studied people infected with the HIV virus and characterized the evolutionary history of HIV viruses with methods that we'll talk about a few lectures from now, if you build an evolutionary tree of HIV viruses compared to SIV viruses, so the H stands for human, S stands for simian. So it turns out that the HIV viruses, 1 and 2, which cause AIDS, do not group together with each other in the tree to the exclusion of SIV viruses. So in fact, HIV-1 is more closely related to some simian immunodeficiency viruses than it is to HIV-2. This is really important for many features like drug treatment of people who are infected with HIV. It matters which strain of HIV they are infected with because they are quite distantly related from each other. And this is a common sort of phenomenon when we're characterizing biological diversity. We don't just want to build trees in order to understand the history of organisms. We frequently want to build trees in order to understand their biology, to make a prediction about something about the organisms, to understand which organisms might have the highest rate of photosynthesis, or which organisms might be good models for studying human biology, and so on. The groupings in the tree help tell us that. So the first thing I'm going to do is go through an example of a term that you will hear throughout the class, which is monophyly, or monophyletic groups. A monophyletic group, I've defined it here in two ways, is a group of organisms consisting of a single node in the tree. So go anywhere in the tree and pick a single node in the tree. And then take all of the descendants of that node, and the node itself, and nothing else. That is a monophyletic group. That's, in essence, the way I've been talking about this, is working from the past to the present. We can also think of it again from going from the present to the past. Take a group of taxa, find their most recent common ancestor, and make a grouping of those taxa, the most recent common ancestor, and all of the descendants of that common ancestor. And you have a monophyletic group. These are also called clades. And, text definitions may be painful, so I'm going to show you what it looks like on a tree. So again, taking this model tree, I've just rotated some of the branches, but it's the same tree. Consider node K in this tree. The monophyletic group that I have labeled there in yellow is the one that includes that node K. So it's node K and all of the descendants of node K and nothing else. That is a monophyletic group. So in the HIV-SIV example, the two HIV strains are not a monophyletic group with each other. And I'll come back to that in a minute. This makes sense? Yeah, question? Yeah, so the question is, can a monophyletic group be any size? Yes, absolutely. Take any node in the tree, anywhere in this tree, and include that node and all of the descendants of that node. And I'm going to show you that in 1 second, the rest of the monophyletic groups in this tree. In some classes, in some books, in some discussions of evolution, people even include the single taxon that is at the tip of a tree as its own monophyletic group. We're not going to do that here. We're talking about multiple taxa for whether or not they are a monophyletic group. So here's another test clicker question. Let's see if everybody has the same problem as before. I'm just going to start the timer here. How many monophyletic groups are shown in this tree? 1, 3, 5, or 7? And answer with the letters A, B, C, and D. And again, you can talk to your neighbors, doodle, whatever you want to do. And usually we'll give, a minute or so to answer some questions. Some that are harder, we might give you a little more time. Some that are easier, we might give you less time. It's not working, or is it? Yeah. There should be a check mark now. Yeah. It says brilliant. All right, everybody done or close enough? Woohoo! Good job, everyone. So the answer is D, 7. And the simplest way to do this is to just go through all the nodes in the tree and count the number of nodes. The number of nodes equals the number of monophyletic groups. So this gets to the question that was asked before. There's one, another one, and so on. And you include the whole thing can be a monophyletic group, including the root node in the tree. So that's how you find monophyletic groups in a tree, yeah.
So technically you can say that HIV1 and 2 are in the same monophyletic group.
Yes, you could say that if you included all the SIV viruses, but you would not say HIV1 and HIV2 are monophyletic unless you said everything is sort of relative to the group of organisms that you're looking at. So because there are other taxa in that tree in between HIV1 and 2, the only way that they are monophyletic is in terms of immunodeficiency viruses in general are monophyletic. But HIV1 and 2 are in fact not monophyletic with each other because there are SIV viruses in between them. Does that make sense? And I'll show you another way of thinking about that. So this is a very common question in evolutionary studies. You'll see this when we talk about plant evolution and animal evolution and microbial eukaryotic evolution. Someone will ask in a paper or in class, are protists, the single-celled eukaryotes that are not fungi basically, are they monophyletic? And we'll talk about whether or not they are. Someone will ask, are sponges monophyletic? And this is basically what you would ask, the same thing as with HIV. You look at whether or not they share a common ancestor with each other to the exclusion of everything else. And since HIV1 and 2 do not share a common ancestor with each other to the exclusion of SIV viruses, they are not monophyletic. But immunodeficiency viruses broadly are monophyletic. So now we just switch sort of gears here, and I just want to show you examples of non-monophyletic groups, because many times people will want to refer to something as a grouping, but it's important to understand that it may not be a grouping that corresponds to a monophyletic group or a clade. And really, a non-monophyletic group, it's sort of obvious, but just to make sure you all understand, it's anything that is a grouping of taxa that is not a clade. So here's an example of one kind of non-monophyletic group. It's a paraphyletic group. And a paraphyletic group is a grouping of organisms where you include the most recent common ancestor of those organisms. So A, B, C, and D, we have a grouping, we include the most recent common ancestor, but some of the descendants are not included in that grouping. So for this to be a clade or a monophyletic group, we would have to, and include A, B, C, and D, we would have to include E, H, G, and F in that grouping for it to be a true monophyletic group. Since we've left out E, H, G, and F, this is a non-monophyletic group. And an example of something like this would be, as you will see later, reptiles. So it turns out that the term that we use to refer to reptiles includes a lot of organisms that the tree might look like this, A, B, C, and D, and then one branch coming out of the middle of reptiles that corresponds to birds. Now this drives some evolutionary biologists crazy that you would have a group with a name that is not monophyletic, but everybody refers to these things as reptiles, and that's just the way it is. But as you will see when we talk about animal evolution, birds emerge from within the middle of the reptile group, which is not the normal way we're supposed to sort of name things in terms of monophyly. You have a question or no? Another type of non-monophyletic group is a polyphyletic group. And a polyphyletic group, usually they're very clearly different. These aren't as clearly different when I've showed it here, but a polyphyletic grouping is a group of organisms where you don't even include the most recent common ancestor of those organisms, and you also don't include some of the descendants of their most recent common ancestor. So an example of this might be flying organisms. Birds, bats, flying insects, flying foxes. Many people are interested in sort of the mechanistic details of flying, and they might be studying the speeds of flying and the wing shapes of flying organisms. They might want to study something about that grouping of flying organisms. But they are not including in that grouping the common ancestor of all insects, bats, birds, and flying foxes and other such things. And they're not including many of the descendants of those common ancestors. And this is what's called a polyphyletic grouping. And we will see examples of both of these, paraphyletic and polyphyletic groups, throughout the class. The most important thing is to be able to look at a grouping. If someone told you, are A, B, C, H, and G a monophyletic group? You should be able to figure out pretty quickly that they're not. Whether or not you can distinguish paraphyletic from polyphyletic is much less important than figuring out that something is monophyletic. And when I say much less important, that means that you're much more likely to get a question about the more important thing on a test. So again, let's just look at this in terms of groupings of organisms. If someone asks you, are G and H monophyletic? You trace to their most recent common ancestor, and you can make a grouping that includes that most recent common ancestor and G and H and no other taxa. So therefore, G and H are monophyletic. Are H and F monophyletic? No, right? Because if we tried to make a grouping that included the common ancestor of H and F, which is down here, we couldn't make that grouping and not include G. So H and F together are a non-monophyletic grouping. I'm not going to do this as a clicker question, but let's just sort of quickly go through this. Which of the following of those groupings on the list on the right are not monophyletic? E, H, G, and F, D, E, H, G, and F, B and C, A, B, C, D, E, H, G, F. Right, so you just sort of quickly scan through and see if you can go back to their common ancestor and make a grouping that includes just them by tracing out from the common ancestor, and you can't do it for that grouping. So now what I want to do is, are there any questions about this? So now what I want to do is go into other issues that are very similar in concept to monophyly and groupings in a tree, but they relate to terminology that you're going to see in the rest of the course, and I just want to make sure you understand this terminology and how to think about it before we drop these terms throughout the rest of the course. So the first one is relatedness. This is very, very commonly used in our course, throughout evolutionary biology, throughout any type of biological study. And by relatedness, what we mean here is phylogenetic relatedness. You know, not first cousin, second cousin, third cousin type of thing, but relatedness of individual species to each other, for example, or phyla to each other. And it's assessed just like we do with monophyly, but it's assessed by examining patterns in a phylogenetic tree. And the way you will see this commonly is to see a question or a statement like this. Is taxon E more closely related to D or to G? So taxon E is more closely related to G because we can find a common ancestor that E and G share that is more recent than the common ancestor that E and D share. So here's the common ancestor of E and G, and that is more recent in the tree than the common ancestor of E and D. That's how we determine phylogenetic relatedness, by tracing along a tree and seeing where organisms share a common ancestor. Yeah? Yes, sorry. It's not really a definition, it's more of an operational statement. So relatedness is, in essence, what we're talking about is how recently organisms share a common ancestor with each other. And it's dropped in lots of biological literature and in the book, etc. And what it means is what I showed you on the tree. So again, if we look at the human and chimp tree, humans are more closely related to chimps than they are to gorilla. Humans are more closely related to gorilla than they are to orangutan. And just as a reminder, if we rotate the tree 90 degrees or if we spin any of the branches, it doesn't change anything in terms of relatedness in the tree. So spinning branches around a node or rotating the tree 90 degrees, the patterns of relatedness are identical. Nothing has changed. So another really important grouping of taxa in trees relates to in-groups and out-groups. In-group is this really simple sort of operational definition, which is if you are interested in studying or showing the evolutionary history of a group of organisms, the in-group is the group of organisms that you want to study. That's it. So if you're studying beetles and you want to study their evolutionary relatedness, your in-group is beetles. And very important is this concept of an outgroup, as we will see in the next few lectures, but I wanted to define it here when we're talking about groupings in a tree. So an outgroup is any taxa or taxon that branches off in an evolutionary tree prior to the existence of the common ancestor of the in-group taxa. So here's our in-group. Here's prior to the existence of the most recent common ancestor, which is right here of the in-group. So these two branches branch off prior to the existence of the most recent common ancestor of the in-group. And they are, by definition, outgroups compared to the in-group. These are always relative. So the outgroups that I'm showing here could be in-groups for another study. If you were studying the evolution of M and its relatives, M would not be an outgroup compared to itself. Everything is relative to each other. So you have an in-group, and then you have to pick an outgroup relative to that in-group. With the primate tree, orangutan is an outgroup of the three most commonly studied human or human-like primates, humans, chimps, and gorillas. Mouse would be an outgroup to the whole clade here. Something that branched off prior to the existence of the common ancestor of whichever you pick as your in-group. So if humans and chimps are our in-group, then gorilla or orangutan could be the out-group, or mouse or squid or anything that branched off prior to the existence of the common ancestor of humans and chimps. Yeah?
So the in-group is monophyletic?
The in-group is not always monophyletic because sometimes people may leave out something in the middle of the in-group. So if you were studying the evolution of reptiles and you left out birds, so birds is this clade in the middle of the reptile group, you could still treat reptiles as your in-group. You would find their common ancestor, and then your out-group would have to be something outside of reptiles. So if I go back to this tree, we could have had another branch in the middle of this tree here, and it might not have been in our in-group. We would have had a non-monophyletic in-group. But we still, to find an out-group for that particular set, we still look for something that branched off prior to the existence of the common ancestor of that group. They do not have to be a clade. Does that make sense? Okay, so another term that is in essence the same in many ways as the relatedness term, but it's used slightly differently, is this sister term. So sister groups are any two clades, or a clade and a taxon, or two taxa. So any sets of two either groupings or individual taxons that are each other's closest relatives. That is, the two things that you are saying are sisters to each other are the two descendants of a common node. So in this tree, for example, A and B are sister taxa to each other. Or G and H are sister taxa to each other. And then you can say G and H, the clade of G and H, is a sister clade to F. So the grouping of G and H is sister to F. And this is also a common sort of use of terminology to explain relatedness in phylogenetic trees. So what is the sister group of D, E, H, G, F? ABC is the sister group. We might not even say group. We might say what is sister to D, E, H, G, F, but it's the same thing. You go back to the node that's below the node for the most recent common ancestor of the group that you're trying to find the sister of. So you go down here and you find what grouping or taxon comes off of that node. Subtle side point is that by definition, sister groupings or sister taxa originate at the same time as each other. They're descendants of the same node. That node represents a common ancestor that occurred sometime in the past. Once that splitting event happens, that's the origin of these two lineages. That happened at a specific time in the past. We may not know when that is, but it did originate at a specific time, and this will be more important when we talk about integrating with the paleontological data. All right, then the last thing I want to mention in terms of relatedness is a complication relative to everything else I've shown you so far, and this is something called polytomies. So far, we've shown that at every node, there is a branch coming up into the node from the past, and then two descendant nodes that come out of the node representing the split into two lineages. This tree, every node has two descendant branches. However, sometimes we show nodes with more than two descendant branches, and that's called a polytomy. There are two main reasons to do this, and people don't always tell you why. So one reason to do this is if you want to represent ambiguity. That is, for D, E, F, G, and H, we may know that they all group together. That is, they all share our most recent common ancestor here. But we may not know what the order of branching is among them. We may not know if D and E are sister to each other or if D and G are sister to each other. We're representing lack of knowledge, in essence, by this polytomy. A second thing that can be represented by a polytomy, although this is very rare to do, is the hypothesis that you had a common ancestor, and there was literally a split into three or more lineages from that common ancestor. So it's entirely possible that this has occurred in some lineages. You could have an ancestral population, a river comes through in that area and splits the land into 10 different quadrants. It's in essence an instant invention of 10 separate lineages. Usually we don't have the resolution to figure that out, so we don't represent that, but that could happen. So polytomy is either going to represent something we don't know, so ambiguity, or in essence a burst of creation of evolutionary lineages.
Question.
Yeah, question. You can go ahead. Oh, all right.
So polytomy is 3 or more branches?
Yes. A polytomy is 3 or more, more than 3, right, that's wrong. 3 or more branches coming out of a node, but this is correct on the figure here. It says more than 3 branches connect to a node. So 1 is coming from the past, and 2 are coming out of the node. So if you have more than that, if you have a node that has 4 branches coming out of it, it's a polytomy. Does that make sense? It would also be a polytomy because I'm counting the branch coming in from the past. Okay? All right. Another question? Yeah.
What happens if you have a node where you have more than one branch coming in from the past?
If you have a node when we have more than one branch coming in from the past, we don't usually represent that, but we do represent crossing of branches, which we will talk about in a week or so.